A research team with roots at the University of Illinois Chicago has demonstrated a CRISPR-based method that programs sequential edits in human cells, rather than conducting all edits simultaneously. This approach aligns more closely with natural gene activation, in which cellular development proceeds step by step. The team used specialized guide RNA molecules, called proGuides, to trigger a daisy chain of edits, activating each step in a defined order. In proof-of-concept studies with human-induced pluripotent stem cells, the method efficiently directed cells toward desired fates, with potential applications in regenerative medicine. One promising application is the generation of pancreatic beta cells, which could reduce insulin requirements for some patients with diabetes. The findings, published in Science Advances, build on prior work from University of Illinois College of Medicine, Professor Brad Merrill's laboratory, and are being advanced through the startup Syntax Bio. The method may offer a more scalable pathway to produce therapeutic cell types. Find more stories at today.uic.edu.